Hello students, today we'll be speaking about the topic organization of eukaryotic chromosome. When we speak about prokaryotic and eukaryotic cells, we see that prokaryotic are usually simplest organisms and eukaryotes are complex organisms. The other difference also holds true. In prokaryote, we have genetic material in the form of a nucleoid, but whereas in eukaryotic organisms, we have the genetic material organized in a complex structure known as chromosome. In this chromosome, we have double-stranded DNA molecule, and then we have proteins. When we try to stretch this double-stranded DNA molecule in a human chromosome, for example, of a 23 chromosomes, we see that it occupies around a length of 1000 millimeter DNA, wherein this 1000 millimeter DNA can be very difficult, or it is very difficult for them to get organized or compacted into the nucleus of a cell, where cell itself is usually in micrometers. So what exactly happens here is that the length of DNA, that is 1000 millimeter DNA of the 23 chromosomes, or if I have to speak about the DNA of a single chromosome, it usually have a length of around 85,000 micrometer. This 85,000 micrometer DNA, it has to compact in the nucleus of a cell whose size also is once again, it is around 2 or 3 micrometers. So how does it happen? So 2 or 3 millimeters. So how does it happen? It all happens because the DNA of the eukaryotic organisms, they compact, they condense. They compact and condense in such a way that there will be a condensation of 10 to the power of fold of the DNA, where the naked DNA molecule organized to form a compact structure called as metaphase chromosome. When we speak about the composition of eukaryotic chromosome, they usually have nucleic acid 50%, proteins 50%. Nucleic acid, it is 40 to 50% of DNA and 10% of RNA. Histones, proteins 40% and non-histone proteins 10%. How the compaction takes place? It happens in 4 levels of packaging. What are the 4 levels of packaging? First level, conversion of 2 nanometer to 11 nanometer. Second level of packaging, 11 nanometer to 30 nanometer. Third level of packaging, 30 nanometer to 300 nanometer. Fourth level of packaging, 300 nanometer to 700 nanometer. The 700 nanometer is of the diameter of 1 chromatid. So if I have 2 chromatids in a chromosome, the structure now comes to around 1400 nanometer. Let us speak of the first level of packaging. The first level of packaging is the conversion of 2 nanometer DNA to 11 nanometer DNA. The thinner, the slimmer DNA as proposed by the Watson and Crick, where they say the diameter of the DNA is 2 nanometer, will now get converted to 11 nanometer size. It was proposed by Kornberg and Thomas. According to this theory, or we call this as nucleosome model, according to this particular model, we see that the DNA coils around itself on a compact structure known as nucleosomes. So what are nucleosomes? Nucleosomes is the histone octamer around which DNA is coiled. The term nucleosome was given by Audit in the year 1975. So in a nucleosome and histone octamer, we have 4 types of histones where each histones are 2 in number. So the histones are H2A, H2B, H3, and H4. Along with these 4 histones, we also have another histone, a larger histone, where we call it as H1 histone, or we call them as linker histone. What exactly happens in nucleosome model is that your 8 molecules of histones, they form a octamer around which now an approximately 146 base pair of DNA gets coiled up. This slide speaks about the structure of your nucleosome around which you have it, which has histone octamer and a DNA. Histone octamers, 8 histones, 2 molecules of H2A, 2 molecules of H2B, 2 molecules of H3, and 2 molecules of H4. As they get compacted around this, you have the DNA of a length of 146 base pairs which come and coiled up. In between the 2 nucleosomes, we have a DNA where we call this linker DNA. The length of the linker DNA, it varies among the species and cell type, which is around 20 to 100 base pairs. As the eastern octamer is formed, we see that there is the diameter of 11 nanometer. And that is why the diameter of the nucleosome is 11 nanometer. So we say the first level of packaging is 2 nanometer to what? 11 nanometer. So how many nucleosomes are present in a human cell? We see that around each nucleosome, around 200 base pairs are usually called up. And when I speak about the amount of the genetic material in the human cell, it is 6 into 10 to the power of 9. On an average, the number of nucleosomes thereby will be 3 into 10 to the power of 7 nucleosomes. So your nucleosomes, repeating units of these nucleosomes, resemble beads on a string, and this has shortened the length of DNA around 7-foot. The histone octamers organize themselves usually in the form of a dimer first and then tetramer, and once again they form a complex of 2 tetramers. H3 and H4 histones, they first organize to form H3H4 dimer, and then to this one molecule of H3H4 dimer, another molecule of H3H4 dimer will come and bind to form H3H4 tetramer. To this, on the other side, H2A and H2B also form a dimers. Now, this two molecules of H3H4 dimer, one molecule of H3H2B dimer, they form a complex. As this are getting complex, another molecule of H2A and H2B dimer comes to form your histone octamer. As this is happening, DNA come and coils around them. The interaction of DNA with the nucleosomes are usually involving hydrogen bonds, salt bridges, nonpolar interactions, and the interaction of the DNA with the histones. And if the interaction is less, that part of the DNA is usually called as the DNA which is having active genes. And the interaction here, if they involve more histones, then that part of the DNA are usually termed as less active genes. This slide speaks about the experiment which was done to speak about what are the components of nucleosomes. A nuclease enzyme from Micrococcus bacteria, they were able to release nucleosomes, and each nucleosome, when they were subjected to the salt solution of high salt concentration, that dissociated the DNA and the protein separately. So DNA, it came as a 146 DNA length, and the octamers are usually like Eastern octamers. Moving on, let us now speak about a second level of packaging. When I speak about the second level of packaging, it speaks of the conversion of 11 nanometer to 30 nanometer. This is usually happened by the arranging of the nucleosomes either next to each other or in a zigzag fashion, where what happens is that the nucleosomes, they are trying to arrange them in the form of one turn, where in each turn, one turn will be having 6 nucleosomes. And usually it happens in G2 interface with the involvement of the histone known as H1 histone, also known as linker histone. Now, the coils of the nucleosomes which are arranged next to each other or in a zigzag fashion, we call it as 30 nanometer chromatin fiber, and it represents the second level of packaging. Now, the arrangement of these nucleosomes in a coil or in a zigzag fashion gives rise to two types of model. One is solenoid model, the other one is zigzag model. In the solenoid model, what exactly happens here is that we see that the arrangement of nucleosomes happens in an helical pathway, and the eastern cores, they arrange themselves sequentially. And this is also called as one-start model. Whereas in the other zigzag model, we see that two opposing nucleosomes, they create a structure where the alternate histone cores become interacting partners. For example, 1, 3, 2, and 4. This zigzag model is also considered as two-start model, and they are also of 30 nanometer diameter. We see that now in the second level of packaging, your 11 nanometer of nucleosomes, when they arrange themselves, they organize themselves either in a zigzag fashion or in a helical fashion. They form 30 nanometer structure. The third level of packaging speaks about 30 nanometer of fiber getting converted to 300 nanometer of your structure. And this happens because of the proteins known as scaffold proteins, which will now also open up to two important terms known as loops and domains. We see that The 30 nanometer, that is second level of packaging, that fibers, they are now thrown in the form of loops on a structural framework, and that structural framework is usually scaffolds. The scaffolds are the non-histone proteins. On the scaffold non-histone proteins, the 30 nanometer fiber, they throw them in the form of loops, and we can see on an average 30 to 90 loops. And in each loop of a 30 nanometer fiber, you might have 180 to 300 nucleosomes, and which covers around 85,000 base pairs of DNA. So now that is the compaction happening. The protein scaffolds, which act as a structural framework for the 30 nanometer fiber to throw themselves in the form of loops, are usually SATB1 proteins, ARBP proteins, A and B lamines of the nuclear envelope, NUMA protein, that is nuclear intermediate filament type protein, and topoisomerase protein. Thank you.